All right, our next speaker is Nidhi Sahni from MD Anderson Cancer Center, and we're going to look at annotation of somatic and other mutations in cancer.
So first of all, I would like to thank the organizers for giving me this opportunity. I guess I'm a little bit different here, coming from a cancer center, talking about somatic mutations. As many of you know, obviously with the advent of next generation sequencing technologies, there are a large number of these genetic mutations that are being identified as we speak. There's been a large set of data sets that are coming out of the Cancer Genome Atlas, the ICGC consortium, as well as UK Biobank and several other data sets that are available. What is lacking is, even though a large number of these cancer oncogenes and tumor suppressors have been studied for decades, a large fraction of the mutations that have been profiled the efforts have been largely lacking. And so here I'm just plotting data that we had compiled for some of these key oncogenes and tumor suppressors for the state of mutation characterization for some of these known oncogenes and tumor suppressors. And as you would appreciate, a large fraction of these mutations, as shown in the gray part of the pie, still largely remain uncharacterized. And so as part of this effort, we started using our technology that we've developed to really look at mutations coming out of the Cancer Genome Atlas, and as well as individual panel sequencing of patients at MD Anderson. And essentially what we do is really different from pool screens, however, doing arrayed screens here, assembling a library of about close to 95 wild-type key oncogenes and tumor suppressors in cancer, and assembling a barcoded library of both mutations and wild type. And we largely characterize the effect of these mutations on two different assays. One is what is looking at the ability of the mutation to confer selective growth advantage to two cell lines that we are looking at here. One is BAF3, which is a murine cell line, which is IL-3 dependent, and MCF10A, which is a non-transformed breast epithelial cell line, which is dependent on EGF and insulin, and classify mutations for their ability to be able to confer a selective growth advantage to either of these cell lines. And then as a second assay, we are looking at protein expression alterations using the RPPA assay that was used universally for looking at protein expression alterations in the 10,000 TCGA tumors. And so just to summarize a large body of work here, just with an example of EGFR, as you can see, we had really looked at mutations along the entire breadth of EGFR. And this was, again, an array screen. And we compare our results, which is shown as a consensus call, compared to a large number of computational algorithms that are used largely in the field to predict the impact of mutations on phenotype or fitness. And as you can see, that our consensus call was largely better than most of the other algorithms, with the exception of this one algorithm that was actually using our training data, so it's somewhat biased. Whereas some of the other oncogenic callers that are used in the field to call which mutations are activating or not largely misclassify some of these mutations. So here again, showing that mutations that were largely activating, for example, in cases where we could model these on the three-dimensional structure, we found that activating mutations largely cluster in the three-dimensional structure of the protein. And so we have assembled this enormous database for this data set here, and I'm very happy to talk to people offline about this FASMIC resource where we've collected the data both from computational algorithms as well as our functional assays. And then in a very quick second here, we are also looking at another kind of genetic alteration here, which are genetic fusion events. And I just got recently funded to build this large library of genetic fusions across cancer. And we are trying to capture both largely recurrent fusion events. And these are events like BCR-ABL that happen in leukemias, but looking at the wide spectrum of different cancers and cloning and assembling the libraries of these large genetic fusions. And then we are essentially using these libraries of these genetic fusions to look at several different assays, looking at cellular localization alterations, phase separation alterations, as well as growth effects. And with that, I would largely summarize that we have a large number of projects in the lab today. I've only talked to you about different assays that we're looking at studying the impact of these driver mutations in cancer. We also have projects looking at mutations in the context of DNA damage networks, as well as identifying novel synthetic combinations using CRISPR-Cas9 screening. And with that, I would like to thank my lab members as well as our funding, and also that my lab is recruiting, so please feel free to reach out if you're looking for positions. Thank you.
Hi, that was a nice talk. You showed some experiments in a breast cancer line and also in BAF cells. Some of the mutations you were studying, though, are generally associated with other types of tumours. So what role do you think context plays, and do you think that might influence your false negative rates, for example?
Yeah, that's a really good question, and I'm asked that always. The thing is, like, of course, as you can imagine, our screens are arrayed screens, and they're quite intense. So we only chose a subset of contexts. But of course, at the Cancer Center right now, we're expanding these libraries to look at relevant tumor types. So we are expanding this to other, for example, colorectal cancer cell lines and other relevant cell lines. But that's a good partnership with the community, actually. So we make these clones available and want people to actually make use of these collections too.
Yeah, I mean, where it might be particularly interesting is in normal cells, for example.
Right. Yeah, that too.
Where the mutations occur early on.
Yeah, absolutely.